Hi, my name is Helen, and I'm presenting on the effect of the C392X mutation on the interaction of thyroid hormone receptor alpha 1 with exportins 5 and 7. Now, to begin, I'm not sure on your biology background, but that might sound like complete nonsense. But don't worry, moving forward, I will try to break things down in a way that is as easily understandable as possible. So to begin, what are thyroid hormones and what are thyroid hormone receptors? Well, just to be very simple, thyroid hormones are messengers that can be sent throughout your endocrine system in order to lead to certain responses within your body. And thyroid hormone receptors receive these messages and can then actually carry out actions. So the thyroid hormone in particular is really important for proper metabolism, for example. And so some people have a condition called resistance to thyroid hormone, in which their body produces the appropriate amounts of thyroid hormones, but those hormones don't interact properly with the receptors. It's like they're not receiving any message at all. And those patients can exhibit a variety of symptoms, including anemia and epilepsy even, or growth conditions. Now, there are a lot of different versions of the thyroid hormone receptor, but moving forward, I'm going to be just talking about one particular version called TR alpha 1, thyroid hormone receptor alpha 1. And one of the mutations that can contribute to resistance to thyroid hormone is a mutation in TR alpha 1 called C392X. And again, that might sound complicated, but really it means that it's just missing a part at the end. And I'll show you right here. Yes, so it is missing this helix 12 section right here. Now, here's another question. What are exportins? I haven't mentioned them yet, but they're these proteins that help, and as a note, proteins are sort of like little cellular machines. So exportins are proteins that help other proteins, in essence, leave the nucleus of the cell. And the nucleus of the cell is this region here where the DNA is. The cytoplasm, in contrast, is where a lot of other parts of the cell exist. Now, our thyroid hormone receptor, our TR alpha 1, can be found mainly in the nucleus, but also to some degree in the cytoplasm. However, it moves in and out, it shuttles. And to move out, it needs typically those exportins to help it get out of the nucleus. And those exportins include exportins like exportin 5 and exportin 7. Now, the C392X mutation that I mentioned is missing a part at the end of the protein that is believed to interact with exportins. So if in a normal protein, the exportin can attach to this helix 12 region and help it leave the nucleus, in this mutant, the C392X mutant, it can't interact with exportins, and thus we think, or I hypothesized initially, it wouldn't be able to interact with exportins and leave the nucleus properly. That meant that if I put my mutant and an exportin in a cell together, we would end up really with more of the mutant in the nucleus than we would otherwise expect, because it can't interact with the exportin to leave the nucleus. So essentially, that's what I did in my experiments. I took my normal, which is my wild type TR alpha 1, and I put it in human cells alongside either just an control or my exportin 5 or 7. And I did the same thing with my mutant with a control or my mutant with exportin 5 and 7. And then under a microscope, I examined the cells to see whether there was more of the protein inside the nucleus or inside the cytoplasm. And that was conducted because, or I was able to do so because our proteins were all tagged with these fluorescent markers. And so I could see, okay, there's more of the fluorescent marker inside the nucleus or more in the cytoplasm. However, ultimately what I found was that there was no impact. With exportin 5, this is really the key takeaway right here. When I had my mutant and the control, or my mutant and the exportin, the localization where my mutant was found was the same in both. And that is illustrated a little bit more clearly here in these images. And the same ultimately was true with exportin 7. Between the control and the exportin 7 conditions, there wasn't a change in where I found the C392X. Version of TR alpha 1. And again, here's an image that somewhat illustrates that. So really the final takeaway is that maybe this exportin sequence, this area at the very end of our TR-alpha 1 doesn't interact with exportins 5 and 7. And thus, when people have this mutation, the reason that the C392X mutation contributes to resistance to thyroid hormone is not tied to its ability to interact with exportins 5 and 7. Thank you, and here are some of my sources. And again, thank you to Dr. Allison, to Vinny, our lab manager, and everybody at the Allison lab for all your help, as well as the Charles Center for funding.